Let's start with denaturing gradient gel electrophoresis. What we're trying to do is to get a pattern, a banding pattern from ribosomal sequences. That is, you do a PCR reaction and recognize that, the products of that reaction is a collection of sequences. Some abundant, some not so abundant. The problem is, if you just run those out on a gel, you get a single band, right? Maybe it's fuzzy because you've got some variation in size, but that variation in size is small. You essentially get one blurry band on a gel, no resolution. Could there be a way, other than size, in which you could resolve those different sequences? And the answer is yes. Because different sequences, even if they're the same size, will have different resistance to denaturation. Different melting points is what that amounts to. And so one way to resolve those is to create a gel. This is a little bit difficult to understand. We're looking at different time points as you run the gel, not different lanes on the gel. You make a gel that is a gradient, where at the top it's a standard gel. But as you move down the gel, you get to increasing concentrations of some denaturant, something that's trying to pull the strands of your PCR product apart. Typically, people will use urea and format it for that. And so these are nitrogen-containing things, right? They interfere with the hydrogen bonding between GC and AT. And so as the concentration of these two chemicals goes up, there's a tendency for them to interfere with that base pairing and cause the things to come apart. There's a big difference between how double-stranded DNA and how single-stranded DNA migrates through a gel. Double-stranded DNA basically forms, I've got my pen here, a more or less rigid rock that will point toward the electrodes and pass through the gel in kind of a streamlined fashion. And the result, it does have to wiggle and twist and that kind of stuff as it goes through the matrix. But it's fairly efficient. It migrates fairly efficiently through the gel because it's rigid and it can take that conformation as it moves through. Single-stranded is like a spaghetti noodle. And as it tries to push through that agar mesh, it gets tangled up in there. It can't efficiently wind up. And so it moves very, very slowly through the agar or polyacrylamide mesh. So what happens then is if you've got a collection of molecules, well, let's start with a single molecule. If you've got a single DNA sequence and you load it onto a gel with a gradient to denaturants, it will move down there as a double-stranded molecule until it reaches the concentration of those denaturants that causes the two strands to come apart. When the two strands come apart, those two strands essentially almost completely stop moving through the gel. And so they're trapped at that location. If you have a collection of molecules, each of them will move down the gel, and when they reach their denaturation point, they'll stop. And so you end up with a pattern where these are the most resistance to denaturation, and these are the least. And that pattern, each one of those bands is a different sequence. You can use the pattern itself to look at the population. And you can look at bands appearing and disappearing in different related populations. Or, if you're interested, you can go back the old-fashioned way and just cut that out with a razor blade, amplify it back up, and sequence it. Find out what it is. Now, you're only going to be looking at the major components of the population, right? Something where there's enough molecules that you can see it on a gel. There are a wide variety of permutations of this. One of them is you can buy a machine that actually creates a temperature gradient across this gel surface. That's not denaturing gradient gel electrophoresis, but temperature gradient gel electrophoresis. It's most common for the PCR primers in this to have long GC clamps at the end. In other words, long stretches of G on one strand, long stretch of C on the other, so that's so resistant to denaturation that it doesn't denature. But since that's only at one end, what happens is that when it does denature, the rest of the molecule denatures. The clamp keeps the ends together, and now you have a single molecule of twice the length. That causes it, one, to stop completely. It doesn't move at all after that. And secondly, it prevents minor migration of those two single strands from separating out and causing doublets of all the gel, all the bands. And so, again, the experiment is simple. You take your sample, isolate DNA, do a PCR reaction, and load it onto a denaturing gradient gel and see what pattern you get. And that pattern is a reflection of the microbial population.